Okay, so this chapter is all about nucleic acids and protein synthesis. Nucleic acids. We had talked about nucleic acids back during our organic chemistry chapter. They are a type of macromolecule. We had carbs, lipids, proteins, and nucleic acids. We did not talk much about the nucleic acids during that chapter because I said we would talk about them later in a different chapter. Well, we are finally here at that different chapter, so welcome. Nucleic acids have two functions. The first thing they do is that they determine inherited characteristics, and the other thing they do is control protein synthesis. There are two types. There are DNA and RNA, and both are found in cells. Little talking about viruses here and there. Either DNA or RNA are in viruses, but never both. So DNA is the first one we're going to talk about, and that stands for deoxyribonucleic acid. It is shaped like a ladder that has been twisted and bonded into the shape of a spiral, as you can see here on the left and right. We call this spiral ladder or twisted staircase a double helix. So that is the structure. Well, what that structure is made up of is nucleotides, which we mentioned in that past chapter. But a nucleotide is a monomer, so it's a very small part. And we're going to take many of these monomers and string them together to form a polymer, which would be our entire DNA molecule. And nucleotides have three parts. They are made up of a phosphate, a sugar. In the case of DNA, that sugar is deoxyribose, and a nitrogen-containing base. And there are actually four options. You have your purines, which would be adenine or guanine, and your pyrimidines, which would be cytosine and thymine. All right, so every nucleotide, every monomer that makes up a DNA molecule will have a phosphate, a sugar, and a base. We're going to be doing a lot of writing in this chapter, and like I said many times already to you, it is not hard, it's just tedious, and these are the shapes that we're going to be using. So for a phosphate, we're going to use a circle with a P. For a sugar, no matter what the sugar is, we're going to use a square with an S. And then we have specific shapes for our bases: adenine, guanine, cytosine, and thymine. And you can see here that we're actually just using the first letter of the name because we don't want to have to write out the actual names over and over again. That would be exhausting. And it's exhausting enough for what we're going to do. So just the capital, make sure, letters of the first letter of the name. Okay. So you have four possible nucleotides in DNA in all organisms. So anything that's living has DNA, guys, or was once living. So here you have, this is an adenine nucleotide. So here you got your phosphate, your sugar, and your base, which is adenine. You have your phosphate, sugar, and base, which is guanine. Phosphate, sugar, base, which is cytosine. Phosphate, sugar, base, which is thymine. So these are your four possible nucleotides. Now, because DNA is double-stranded, you could have molecules on the left or the right, depending on which side you're looking at. So the reason we're showing these to you flipped around is to show you that these are the same nucleotides. This is still an adenine nucleotide, but this is what it would look like if it was on the other side of the DNA molecule. So just keep that in mind. So these are your four monomers, your four types of nucleotides that we're going to use to build a DNA molecule. All right, so here's the start of building this molecule. Here you'll see I still kept my four nucleotides here. You'll see that each base is attached to a sugar and a phosphate, sugar and a phosphate, sugar and a phosphate, sugar and a phosphate. That's good. You'll also see that we added a bond here between the nucleotides. You don't see that here, but here you do. So the reason for this is because we don't want individual nucleotides. We want them chained together because that's what's going to start to create that DNA molecule. So you make the bond from sugar to phosphate of one molecule to the next, sugar of one nucleotide to a phosphate of the next, sugar of one to the phosphate of the next, and you go all the way down your molecule. So that's how you do the one side of the molecule. In this case, it's our left side. If we look over to the right side, we use base pairing rules. So you'll notice that the shape of A fits very nicely into the shape of T, and G fits very nicely into C. These are our base pairing rules, guys. A is always going to pair with T, and G is always going to pair with C when we're talking about DNA. The saying for that, to remember that, is apples in the tree, car in the garage. Okay, A with T, C with G. Always for DNA. So we use our base pairing rules so that we can create the right side of the molecule, A with T, G with C, C with G, T with A. Each one of these bases needs a sugar and a phosphate. And then we need to connect those with our sugar to phosphate bond to make a chain. Now that we have the left side and we have the right side, we need to combine them in the middle. And we do that with hydrogen bonds. Now, because of the chemical makeup, adenine and thymine get 2 hydrogen bonds, and guanine and cytosine get 3. So you'll go down your entire molecule and you'll fill in these bonds. It's the same every time. C and G get 3, T and A get 2. Okay, so here's the chemical makeup of it in case you're wondering. So here's what adenine looks like chemically. Here's what thymine looks like chemically. So here's where these bonds are able to form. So that's why there's 2 here and 3 there, and why they're complementary. Okay, which brings us to worksheet 1. We've already done this in class at this point, well, most of us anyway. I'm going to now play a video of me doing this by hand. So those of you that missed it will be able to catch up a little bit. Stay tuned. Okay, so here we go with worksheet 1. You'll see here that we have the bases of a DNA molecule here. So we now know that every nucleotide has a phosphate, a sugar, and a base. So here we have T, and I'm filling in the sugar and the phosphate. Okay, so you got to fill that in for every single base going all the way down. Base gets a sugar, and it gets a phosphate. Cytosine is going to get a sugar, and it's going to get a phosphate. Guanine gets a sugar, and it gets a phosphate. And you'll notice that I have to fill them in all the way down. Okay, so I just fast forwarded a little there. So now you'll see I have individual nucleotides, and I just need to connect them together. So I make the sugar to phosphate bond going all the way down, and this is going to create a chain of nucleotides, which is what I'm looking for. Good. So the left side of the molecule is now completed, so I got to move on to the right side. I'm going to use my base pairing rules. I'm going to draw in my shapes here. And I know that T pairs with A, and A pairs with T. G pairs with C, and C pairs with G. So I just go down the entire molecule and fill in those shapes and in those letters. So I go all the way down. So I've done that here for you. We filled all that in. Now every base that we just created needs a sugar and a phosphate. So I once again have to go down the entire right side of the molecule now and fill in those sugars and those phosphates. Okay, so I'm going to fill those in. There we go. And now I just need to make a chain by going sugar to phosphate all the way down the molecule. Once again, this makes a chain of nucleotides, which is very important. Good. The last thing I need to do, since I had the left side and the right side, now I need to go through and connect the middle. So there are hydrogen bonds we mentioned earlier, and there are 2 between adenine and thymine, and 3 between guanine and cytosine. So I just go down the molecule, and where I see C and G, I put 3, and where I see A and T, I put 2. After that is done, the molecule's done. Worksheet 1 is complete. That's all you need to do. So as you can see, it's definitely not hard. It's just tedious. Okay. Once you get the hang of worksheet 1, you can move on to worksheet 2. There are 2 on the front and 1 on the back. The one on the back you have to correct and complete, so there are issues on there. You must be able to understand worksheet 1 and 2 before we can move on. The idea is that this chapter will keep building upon itself. So this is the very basics, very tedious, but not horrible. When we move on to worksheet 3, it will move up a step and we'll start doing something a little different. So we're going to stop the video here today. And when we move on to the next topic, we'll get a new video. Thanks.